Welcome back to the deep dive. Today, we are not just looking at disease, we're looking at the body's ultimate, brilliant, and sometimes, well, brutal survival guide. Ever wonder why a bodybuilder's heart grows larger but doesn't create new muscle cells?
Right.
Or maybe why the lining of a smoker's lung totally changes its identity?
Yeah, those are great examples.
We're peering inside the cellular architecture today to uncover the 4 fundamental survival trades our cells make when they're facing stress, whether that's overwhelming demand, vanished resources, or, chronic irritation. Our mission to quickly and thoroughly understand these core adaptations: hypertrophy, hyperplasia, atrophy, and metaplasia.
And this is truly a really important boundary in medicine, isn't it? These cellular adaptations represent the razor's edge between health and disease.
Okay.
When a cell successfully adapts, the tissue maintains viability under stress. But when the adaptive mechanism fails, or maybe if the stress just persists too long, that's when functionally significant pathology entry and potentially even cancer begins.
Okay, right. Let's unpack this then. Let's start with the adaptation that involve increasing capacity. The first 2 strategies are about growth.
Okay, yeah, let's start with hypertrophy. This comes from the Greek for overnourishment, and it's precisely that. An increase in the size of individual cells, which then leads to the increased size of the organ.
Right.
What makes this so vital, though, is that the hypertrophied organ has no new cells. The existing cells simply bulk up. They synthesize more structural components, more organelles, things like that.
And this is the only option for cells that have lost their capacity to divide, right?
Right.
You think of permanent cells like our neurons, or critically, our heart muscle fibers.
Absolutely. The heart is, well, it's the textbook example. It cannot undergo hyperplasia. It can only respond to increased demand by growing bigger cells.
Makes sense.
We see this physiologically, like the massive controlled growth of the uterus during pregnancy. Estrogen signaling causes those smooth muscle fibers to swell up and multiply their internal machinery, to carry the load.
That makes perfect biological sense, meeting a temporary, massive functional demand. But when this adaptive effort moves into the pathological realm, the stakes rise dramatically, don't they?
Oh, they do. The heart under chronic pressure overload, say, from long-standing hypertension or maybe a faulty valve, in must hypertrophy. Initially, this is fantastic. The heart copes.
Yeah, it's adapting.
But if we connect this to the bigger picture, the increased size often outstrips the blood supply. It starts demanding more oxygen than the coronary arteries can deliver. And that's when the adaptation becomes maladaptive. It can lead to degradation, cell death, and eventually heart failure.
But hold on a second. If this is a survival mechanism, why doesn't the heart under chronic stress try to make more cells? I mean, why is hypertrophy the only option, even when that strategy often fails long term?
Yeah, that's a really important question. The adult heart muscle simply lacks the regenerative capacity you see in tissues like the liver or skin. The cell cycle controls are, well, they're locked down.
Oh.
Therefore, the cell's only available genetic blueprint for increasing its workload capacity is to increase volume, not head count. It's a mechanism born of biological restriction, really.
Okay, now let's zoom in on the molecular switch. How does the cell actually know it's overworked? What flips that switch?
It's highly regulated. You have mechanical sensors within the cell that detect the increased workload, literally the stretching and pressure, and they activate these complex signaling pathways.
Okay.
And what's fascinating here is how the type of stress seems to determine the mechanism. For physiologic hypertrophy, say, from resistance exercise, the PI3K-AKT pathway seems key. It often leads to a healthy, balanced growth.
Right, like in athletes.
Exactly. Conversely, in pathologic states, G protein-coupled receptor pathways activated by things like growth factors and vasoactive agents tend to dominate, and that often leads to the more problematic enlargement we see in disease.
And in the pathological heart, there's that incredible phenomenon known as the fetal switch. That sounds like the cell is hitting some kind of reset button.
It basically is. Under prolonged, unrelenting stress, the heart cells actually switch their gene expression. They start producing the slower, more energetically economical fetal isoform of myosin, that's beta-myosin, instead of the adult contractile protein, alpha-myosin.
Wow.
They also increase expression of ANF atrial natriuretic factor. That's a hormone that causes salt excretion by the kidney, kind of hoping to reduce the blood volume and ease the hemodynamic load.
So it's trying to help itself out?
Yeah, it's the cell basically betting its long-term optimal function against immediate survival. It's a huge trade-off.
That's amazing. The cell literally regresses to an older blueprint to survive a modern crisis. Okay, let's move to the second growth strategy then, hyperplasia.
Right. Hyperplasia. This is the increase in the number of cells in an organ or tissue. So this requires cells that are capable of division or active tissue stem cells.
Okay, so different from hypertrophy.
Exactly. They often coexist with hypertrophy, actually.
Yeah.
But hyperplasia is the strategy for tissues that can regenerate and make more cells.
And we see fantastic examples of healthy physiological hyperplasia all the time, right? Hormonal hyperplasia in the glandular epithelium of the female breast at puberty or during pregnancy, or that compensatory hyperplasia that allows for like rapid liver regeneration if a surgeon removes part of the organ.
Yes, exactly. Those are controlled, reversible responses. But the problematic forms are pathologic hyperplasia, often caused by excessive hormonal or growth factor stimulation.
Like what?
Well, a common example is endometrial hyperplasia. An imbalance between estrogen and progesterone leads to overgrowth of the uterine lining, often causing abnormal bleeding.
Okay.
Another really common one is BPH benign prostatic hyperplasia, which happens in many aging men, driven by androgen stimulation.
That brings up a critical distinction, though. Pathologic hyperplasia is still a controlled adaptive response, but we treat it as dangerous. Why is that?
It's because of that increased cell turnover. I mean, even though the process is regulated, it creates what we sometimes call a fertile soil for cancer.
Oh, right.
Increased cell division just naturally elevates the risk of acquiring genetic aberrations, mistakes in the DNA. Every time a cell divides, there's an opportunity for a mutation that could eventually drive unrestrained proliferation. It increases the probability of malignant transformation down the line.
So if the cell's answer to demand is to build either bigger cells or more cells, what happens when resources vanish? Let's turn the dial 180 degrees to the mechanism of shrinkage.
Yeah.
Atrophy.
Right, atrophy. This is the reduction in the size of an organ or tissue, and it's due to a decrease in both cell size and, sometimes, cell number.
Okay.
The cell basically reduces its metabolic machinery, shrinks down to achieve a new, lower demand equilibrium. It's trying to permit survival under conditions of scarcity.
And the causes are tragically common. We've all seen disuse atrophy like muscle wasting after a fractured bone is immobilized in a cast. The muscle fibers decrease in size really rapidly. But what about the more systemic causes?
Well, we see atrophy from diminished blood supply leading to senile atrophy. For instance, the progressive brain atrophy often seen in late life due to atherosclerosis, cutting down blood flow.
Okay.
And then there's the profound wasting condition known as cachexia. That's the extreme muscle wasting you see in severe malnutrition, chronic inflammatory diseases, and cancer.
Right, cachexia.
Cachexia is often driven by the chronic overproduction of inflammatory cytokines like TNF tumor necrosis factor, which actively signals the body to break down muscle protein.
So if resources are scarce, the cell needs to reduce its size quickly. This must require something more urgent than just slowing down making proteins. It sounds like active destruction. How does the cell register that lack of input, and what's the internal biological response to that silence?
Yeah, it's not just passive. It results primarily from a really sophisticated, regulated increase in protein degradation. The cell activates a highly regulated cleanup crew called the ubiquitin proteasome pathway.
Ubiquitin proteasome.
Okay. Think of ubiquitin as like the molecular kick-me sticker, the tag.
Right.
Nutrient deficiency or disuse activates these enzymes called ubiquitin ligases, which ruthlessly tag cellular proteins, especially those in muscle,
Yeah.
with this small peptide, ubiquitin.
Got it.
This tag marks them for wholesale demolition. The proteasome is like the cellular garbage disposal, a barrel-shaped enzyme complex. It recognizes that ubiquitin tag and just ruthlessly grinds up the protein into small amino acid fragments.
Which the body can then reuse.
Exactly.
Yeah.
Reuse them for energy or synthesis elsewhere. That is how the muscle wasting in catabolic conditions like cancer cachexia is accelerated so rapidly it's an active breakdown.
It's an aggressive but necessary triage system. And sometimes the cell tries to clean up structures it no longer needs via increased autophagy. It's essentially eating its own parts, right?
Exactly. Increased autophagy leads to more internal cell debris being packaged up in these things called autophagic vacuoles. If that debris, often it's lipid residue from membranes, resists digestion, it can persist as membrane-bound residual bodies. And we call these lipofuscin granules. When enough of these accumulate, they actually impart a yellowish-brown discoloration to the tissue. That's what we call brown atrophy, which you can sometimes see in the heart or liver of elderly or undernourished patients.
That is a fascinating molecular footprint of scarcity. Wow. Okay, now let's turn to the last and maybe most dramatic survival strategy, the complete identity swap. This is metaplasia.
Metaplasia. Yeah, this is a reversible adaptive change where one differentiated cell type is replaced by another cell type that is theoretically better able to withstand a specific adverse environment.
So it's a trade-off.
It's a core adaptive trade-off, absolutely. The cell sacrifices its specialized function for increased ruggedness, essentially.
And the most common epithelial metaplasia is that columnar to squamous switch, and the classic tragic example is the habitual cigarette smoker.
Right. In the smoker, the respiratory tract is just constantly irritated by the smoke. The specialized ciliated columnar epithelium, those are the cells that secrete mucus and have cilia to sweep away particles, they get replaced by tougher, multi-layered stratified squamous cells.
Like skin cells, almost.
Kinda, yeah.
Yeah.
More durable.
Yeah.
These rugged squamous cells survive the chronic irritation much better. However, the price of this adaptation is steep. The critical defense mechanism is lost. No more mucus secretion, no more sweeping action by the cilia, which makes the patient highly vulnerable to chronic infection.
It's amazing. The cell literally trades its function for toughness. That's a brutal adaptive bargain. And this adaptation, while it's protective initially, carries a huge long-term risk, doesn't it?
It absolutely can. It can initiate malignant transformation. If that chronic irritation persists, the reprogrammed metaplastic epithelium can progress further, potentially leading to tumors like squamous cell carcinoma. This is why metaplasia is generally considered a red flag, an undesirable change, despite its adaptive origins.
And we see the reverse adaptation, too, sometimes. In the GI tract, for example, like Barrett esophagus.
Yes, exactly. In Barrett's, the normal squamous lining of the lower esophagus is constantly bathed in refluxed gastric acid. The cells respond by switching to an intestinal-like columnar phenotype.
Why would they do that?
Well, this switch is adaptive because columnar cells are generally better at handling acid exposure than squamous cells are. But that change is associated with a significantly higher risk of developing glandular tumors known as adenocarcinomas. So again, a risky trade-off.
So this isn't the existing cell just morphing into a new shape. What is the fundamental mechanism behind this identity shift? It sounds much deeper.
It is deeper than just a surface change. Metaplasia results from the reprogramming of local tissue stem cells.
Yeah.
It's not the mature cells changing. They tend to die off.
Oh, okay. The stem cells.
It's external stimuli, cytokines, growth factors, changes in the environment like chronic irritation or acid, they influence the nearby stem cells to express a whole new set of genes. This drives them down a different differentiation pathway entirely.
Interesting.
Well, we know, for instance, that deficiency or even excess of vitamin A or retinoic acid can directly regulate the transcription factors that govern this stem cell differentiation process, potentially causing squamous metaplasia.
Wow. So it's really redirecting the raw material.
Precisely. It's changing the instructions for the new cells being made. So if we just summarize our deep dive here, hypertrophy, hyperplasia, atrophy, and metaplasia are all attempts by the cell to maintain viability when faced with stress, whether that means increasing capacity, decreasing demand, or completely changing identity. They're all fundamentally highly controlled biological decisions.
So what does this all mean for us? I mean, these concepts really illustrate that disease often starts not with some random mistake, but with the failure or maybe the maladaptive consequence of a highly intelligent, controlled biological effort to survive.
That's a great way to put it.
The most fascinating adaptations, like you mentioned, the heart switching back to fetal genes or the lung trading protection for durability, often seem to come with the highest long-term cost.
They often do. Survival now versus optimal function later.
So here's a final thought for you, our listener. We saw how the heart switches back to fetal genes under pathologic stress. If the body's ultimate survival mechanism, when faced with insurmountable stress, is sometimes to regress to an earlier, perhaps more energetically economical developmental state, what other forms of common chronic illness might secretly be rooted in similarly ancient but ultimately maladaptive survival blueprints? Something to think about until next time on The Deep Dive.